My name is Sarah Kenzar, and my final project research topic concerned agricultural runoff and river ecosystems. Specifically, I'll be discussing the Lake Sturgeon adaptations within the Mississippi River Basin. The human domestic system being studied is agricultural river ecosystems in the Mississippi River Basin. Different fish species, like it, develop adaptations in response to water pollutants. A controlled experiment was conducted over a period of several months, employing the testing of water turbidity and dissolved oxygen to do so. The experimental group was exposed to a higher than normal concentration of various pollutants for a comparison to the control group. The following results are presented and discussed in this science poster. My initial hypothesis is that river fish, like the lake sturgeon, may develop behavioral and structural adaptations, likely short-term adaptations, when exposed to sustained high levels of agricultural runoff in a laboratory setting. My objectives for this research project were: one, to observe any potential deviations in the lake sturgeon's experimental group in comparison to the control group, and secondly, to determine if said deviations demonstrate a positive correlation in the development of short-term adaptations, be it structural or physiological, in the lake sturgeons with increased contaminant exposure. My research centered around 4 different areas, beginning with a discussion in environmental science role in my research topic. Environmental science specifically has been used to monitor and manage the agricultural impacts in the Mississippi River through nutrient reduction strategies, integrated pest management techniques, and water quality conservation practices. In-stream water quality monitoring is also utilized through specific initiatives like the National Water Quality Initiative. Said monitoring has been done on both the volunteer level of everyday farmers and citizens, and has also been carried out by scientists with the Environmental Protection Agency and the USDA. Concerning scientific uncertainty, there is a certain amount of that, with the extent to which the anthropogenic origins of water contamination impact fish like the lake sturgeon in the long term, given the prevalence of shorter term studies and the difficulties associated with separating the impact of other factors like pre-existing biological conditions. This is where environmental science creates multidisciplinary solutions by separating the natural from the anthropogenic origins of contaminants in marine ecosystems. My methodology involved a number of steps. First, I installed a turbidity meter at a location in the Mississippi River for 1 month. At the same time, I utilized the optical dissolved oxygen method with a dissolved oxygen meter for 1 month in that same location. Then a number of lake sturgeons were collected from various regions of the Mississippi River and were divided into experimental and control groups. The experimental group was exposed to cloudier, more nutrient-rich water samples over several months, while the control group was exposed to normal samples also over several month period. The experimental group received observation for various physical changes and physiological changes in their gill structure, swimming patterns, breeding habits, and others. An in-depth analysis was done on the changes that were observed in both the control and experimental group. My conclusion: I determined that lake sturgeons in the control group grew at far slower rates in comparison to those of the control group, demonstrated extreme lethargy, developed severely clogged gills, and did not engage in breeding behavior that is consistent in that fish species. The control group, however, developed no new adaptations and largely retained the same characteristics found and observed in the beginning of the study. My recommendations: the results are straightforward and do indeed demonstrate a causal relationship between exposure to higher than normal amounts of water pollutants and short-term adaptations to better be suited towards said unfavorable conditions. Given these results, I believe that the Environmental Protection Agency and various state governments should, in fact, mandate more specific targets in ecologically vulnerable portions of states like Mississippi for local implementation. Less reliance on volunteer efforts is also needed. Additionally, more long-term river quality monitoring projects need to be in place, as many existing projects tend to be short-term and do not reveal the extent to which species like the lake sturgeon can be impacted over a long period of time. Lastly, the tracking of specific endangered fish species like those of the lake sturgeon in various basins across the United States may prove to be instrumental. Taking a species-based approach in this manner would allow for greater focus on particular species that are endangered and face disproportionately higher rates or risks of endangerment and extinction, and will allow scientists to make better use of available resources in studying the impacts of water pollution, ranging from sediment, fertilizers, and soil runoff from agricultural activities in those fish species. Ultimately, and in doing so, the viability of the lake sturgeon can be ensured to continue for generations to come. This in turn would help boost the preservation of fish species like it, while improving upon existing water conditions where they are found.